All right, last time we talked about the structure of DNA, and so now we're going to talk about how DNA replicates. So DNA replicating is said to be semi-conservative, and this basically just means that each strand of DNA is going to break apart and create a template so that these two can be paired up correctly. These two can be paired up correctly, and these two. But that also means that this original strand and this new strand are copies of each other. But when we are making a new DNA through DNA replication, we're not making copies, we're making complements. DNA replication is going to start when we unwind and unzip DNA. We're going to use a special enzyme for this unzipping called helicase. And this is basically going to go through and break the hydrogen bonds between the A and the T, or the C and the G. So break those bonds break the hydrogen bonds, all right? So that's what helicase does. Then we're going to match up our pairs, and we have a DNA polymerase that's going to manage this for us. All right, that's another enzyme. Remember, our pairs, A always goes with T, and C always goes with G. We have a leading strand that's red, that's put together really easily in the 5' to 3', and then our lagging strand is 3' to 5'. Now, DNA polymerase can't do that. So it can't do it that way. It has to go 5 to 3. So we're going to see that the lagging strand is going to end up with what we call Okazaki fragments. And those are going to have to be stitched back together with ligase, which we'll talk about in a minute. And then the last thing we have to do is proofread, and our friend DNA polymerase, he does that as well. All right, so now I'm going to show you a video that goes through all of these steps, and then we'll come back together and talk about these steps.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
All right, so this is kind of a graphic representation of the different steps. So the first step, in my opinion, is we got to unwind it, but they don't really show that part. They do show the unzipping part. And this is number 2, where we create a replication fork. So a replication fork looks just like here's the fork, and then this is one tongue of the fork, and that's one tongue of the fork. And we do that by using that enzyme helicase. So once we've broken all these bonds, because remember the bonds between these two original strands are weak, those are hydrogen bonds. So we're going to break those with helicase. Then we're going to create, so this is up here, this is the leading strand right here, right on this side. And so I'm just going to kind of show you using this side right here. So the leading strand, here is our RNA primer. We got to lay that down. And then what's going to happen is you're going to have let's see if I can get a different ink color here. You're going to have an enzyme called DNA polymerase. And the DNA polymerase is going to start grabbing the matching pairs. So if I have it's going to just start adding pairs to this new thing. So T always goes with A. T always goes with A. G, C, G, G, C, G, A, T, and so on. Okay? So it's just going to start making the complementary base pairs moving in that direction. So that's the leading strand. Okay? RNA by DNA polymerase. Now, on the lagging strand, we have to start on the 5' end because we can only go 5 to 3', and we have to create these little fragments. All right? That leads us to then we have to have sort of like before we get to the proofreading, we have to have another step in which DNA ligase is going to glue these together, so that they will end up being one continuous strand. All right? And then we have our friend DNA polymerase that has to come through here and proofread. So it creates these daughter strands, and then what it does is it comes through and it double checks its work. So the first thing it does is this right here shows you a mismatched pair. So this has a G, I don't know if you can see that. This has a G and that has a T. Well, we know G always goes with C. So this should not be a T, this should be a C. Oh, I apologize. Yeah. This should not be a T, this should be a C. So it's gonna come over here and it's going to make a C right here where there should be a C. All right, and it fixes it right here. So that's a mismatched pair. The next one is it can have added an extra letter that doesn't belong, okay? Or it can have forgotten to add a letter, and so everything is kind of shifted downward. So the mismatch repair are going to remove it and add back in the correct thing. All right. And then it can also repair thymine. A lot of times thymine gets damaged. And so they can come in and use something called a dimmer to sort of fix that up. All right. And so that is DNA replication.